Okay, so this lecture wraps up the one that I was doing this morning. Apologies that it ran over time. As I mentioned before, this is the fusion of two previous lectures, and getting it all within the 75 minutes is tough. I'm trying not to let this run too long, but I want to highlight the fact that although I began by talking about um, the Cambrian Explosion, we didn't actually quite get there in terms of the lecture, because we did what happened in the discoveries of the Neoproterozoic. People were trying to find the origin of the modern diversification of bilaterians, and they looked among the various sorts of Neoproterozoic animals. And although they initially tried to say, oh, here's a proto-trilobite and here's a proto-echinoderm and so forth, they weren't really there. So we have all this diversity in the Cambrian Explosion, and I even showed the phylogenetic diversification we saw, but we didn't get to many of the modern groups, nor did we get to mineralization, except in the case of things like Cloudina and that little spheroid Nomicolathus, probably an early sponge. So this video is going to actually look at what evidence we have of the Cambrian Explosion itself. I do want to point out, too, that the Cambrian Explosion, when people use the phrase Cambrian Explosion, they're really talking about two different things which are not necessarily simultaneous with each other. And this will be an issue that shows up later in this lecture. Classically, the thing that concerned Darwin was the fact we didn't have mineralizing organisms, and then suddenly we did, and when we had them, they were already diversified across a wide variety of forms. So that seemed to him to be very strange, the sudden appearance of mineralizing skeletons. So that's one idea of the Cambrian Explosion, is the rapid appearance of mineralized skeletons among organisms. But there's another take on this, and that is that the Cambrian Explosion is the diversification, the phylogenetic diversification of metazoans, and in particular bilaterians. So that the first one has to do with the shells, the other one has to do with the family tree. And those don't have to be simultaneous events. If you think about it, you could potentially have branching among the major groups of animals, and it goes for millions of years or tens of millions of years or longer. And if they don't all form shells until a later point, that divergence and the formation of shell are widely separated in time. So let's see what the evidence actually talks about. Well, here's our family tree of animals yet again. And in this case, what's mapped onto there is mode of life, and particularly how they get food. So over here we see sponges are suspension feeders. Well, we know that. The ancestral ctenophore, that is the ancestral comb jelly? Well, it's hard to say if they're mostly suspension feeders or predators. Modern ones do both. They have these long tentacles. They're sticky tentacles, not stinging tentacles, and they'll grab particles and pull them in, but there are many ctenophores that are suspension feeders. Same thing for cnidarians, for jellyfish and sea anemones and their cousins. Some are purely suspension feeders, but some use their stinging cells. That's the reason you don't want to get rubbed up against by the tentacles of a jellyfish or a stinging coral or whatever. In our particular lineage, so we are chordates. Chordates are the group that vertebrates belong to. Ambulacraria is the group to which the echinoderms, starfish and their kin, belong to. And the ancestral forms on both sides are suspension feeders, although we've gone on to do many different things. There's a group called the chitinathes, or arrow worms. They're ancestrally predators. On this side of the tree, mollusks are ancestrally grazers, and we saw that in the form of Kimberella. Notice they're the odd critter out here. They're the one group that does something radically different. The ancestral annelids, so segmented worms, suspension feeders. The ancestral brachiopods, suspension feeders. The ancestral flatworms, a combination of either predators or detritivores. Detritivores are organisms that digest the organic material in the sediment. Ancestral bryozoans, in fact all bryozoans, suspension feeders. Priapulids we encountered last time, or earlier today. They are those little worms. They're ancestrally predators. Nematoids, those are nematodes and their cousins, roundworms, detritivores. And then figuring out the lifestyle of the ancestral member of the bugs, of the group called Panarthropoda, is tough. They're so diverse. So maybe predator, maybe suspension feeder, maybe detritivore. But there's one big pattern overall, and that is the ancestral form of life for all metazoans, for all animals, is suspension feeder. And in fact, suspension feeding remains the basic state on the tree throughout, all that way down here, all the way up here. And then it's only over here on this lineage and this lineage that it changes. On mollusks, it goes to grazers, and then it's uncertain if it's detritivore, suspension feeding, or predation in this group. By the way, the ecdysozoa, so priapulids, nematodes, and bugs. Ecdysis is shedding your shell, shedding your cuticle, like insects do. All these groups in this clade actually do that. They get rid of their cuticle as they grow. Now, I mentioned at the end of the live part of the lecture that one possibility for the reason we don't see the worms in the fossil record, that is the vast diversity of lineages down here prior to the Cambrian explosion, is maybe they were just tiny. We're the Ediacaran worms in the meiofauna. So meiofauna, it's not as small as microfauna, but it's not macrofauna. So that's meio means middle-sized. Basically, this is sand-sized organisms. So on this picture, these are drawings of modern meiofauna, and these boulders, those are sand grains. And there's a lot of biological diversity at that size. Now, the reason this is important in terms of fossils is that's a hard size fraction to fossilize. It's hard to preserve. You couldn't preserve these, for instance, in sandstones, and you wouldn't be able to displace the sand. The sand is as big as the organism. Same thing with silt. These things are not much bigger than silt. You might preserve these in mud as carbonized impressions or carbonized films, but that's about it. And indeed, here's a photograph. Actually, it's a composite photograph of a bunch of meiofauna found in some sand. Some of these are actually the juveniles of later forms. So this will grow up to be a much larger clam, as will these ones. And there's a little baby brittle star. There's a little clam. There's a little snail. Some of these, though, are the adult size. And here someone took a picture under a microscope, and we see these very tiny organisms. There's forams for scale. And there happened to be a handful of fossils from the Cambrian that represent clades that are otherwise not well represented in the actual fossil record, like loricans, which are weird sort of suspension-feeding worm, that actually happen to be preserved. And they're part of the meiofauna. And this little thing, this is a reconstruction of it, the fossil is nowhere near this good, is interpreted by its discoverers as being a member of the deuterostome line. That's the line that contains you and me and other vertebrates, as well as the echinoderms in our kin. And the idea is this is something that is stem deuterostome. Now, I talked about how earlier this lecture that the GSSP, the base, I should say, of the Cambrian, labeled GSSP here, was on the first appearance of Treptichnus pedum, or Trichophycus pedum. Well, once that level is formally declared in the scientific literature, that's it, and that becomes the boundary. And it turns out, subsequent to that naming back in the 1990s, people have found Treptichnus pedum slightly lower in stratigraphic section than that. So great, that's life. It also means that we've got priapulids down here. So priapulids are present by the late Ediacaran. As I talked about in class, there are some other trace fossils of priapulids, including possibly those burrows, those punctures into Cloudina shells. Now, the Cambrian fossils include some really spectacular sites. In fact, for many people, the archetype of a Lagerstätten, remember, a site of spectacular preservation, is either the Burgess Shale fauna, which is from the mid part of the Cambrian, or the Chenjiang fauna, which is from China, which is from slightly earlier in the Cambrian. And there are several other Lagerstätten from the Cambrian, from the first half of the Cambrian, which is great because it gives us a window into the biological diversity of non-shelled animals at around the time that shells begin to become common. So let's just review some early Paleozoic, some Cambrian fossils here. Trilobites we'll spend a lot more time on next week. They are a group of bugs, technical term arthropods, who had a skeleton of calcium carbonate. At least the top skeleton of them was calcium carbonate, even harder than the shells of a crab or a lobster today. They were, for a long time, well, they appear in the Cambrian, but not at the very base of the Cambrian, but fairly early on, and survive, as I talked about earlier, to the very end of the Permian. Although the diversity decreases, they are phenomenally important as index fossils. They are wonderful things to look at. We'll talk more about them in class later. And this is showing their diversity radiating out in the earlier part of the Cambrian and surviving till the end of the Permian. Somewhat more prosaic, but still surviving to today and actually having an even better fossil record, are the gastropods. Gastropods are snails, both land snails and sea snails. They have a shell of calcium carbonate, little squiggly soft organism, but with a big shell around them, and they radiate very early in the Cambrian. The clams do as well. The technical name for the group of clams is bivalves or bivalvia, the two-shelled ones. And the echinoderms also radiate out in the Cambrian. Echinoderms are the group that today include sea urchins, sea cucumbers, starfish and brittle stars, and sea lilies or crinoids. But none of those particular groups show up in the Cambrian. Instead, there are earlier stem echinoderms at that time. Echinoderms have a very unusual biology. They are although they're bilaterians, they're not bilaterally symmetrical. And they have a skeleton comprised of little bits of calcium carbonate, which can be fused together or can be sort of just loosely attached. And that's why something like a sea star, a starfish, can move its arms up and down, or a sea cucumber can burrow like a worm. Other ones have more rigid skeletons, like the core of a brittle star or a sea urchin. So both these groups, calcium carbonate shells. There are even stem mollusks, creatures more closely related to crown mollusks than to anything else, but outside the crown group in the Cambrian. There's also the Euconodonta. I talked about how they were survivors, although at low diversity, of the Permo-Triassic extinction in Tuesday's lecture. Conodonts are technically the name for the little elements that are in their mouths. Euconodonta is the clade. They are little micropredators. They were long known for these mouth parts, some stabby ones, some crushy ones, and eventually they were found in association with each other, so we knew they were from the same animal. Now, these are not calcium carbonate. They are hydroxyapatite, that is calcium phosphate, which made people curious from the 19th century onward to say maybe these are teeth, because after all, our teeth and all vertebrate teeth are calcium phosphate, or hydroxyapatite, in which case these would be the parts of fish. However, there was a long debate in the 19th century and much of the 20th century as to where conodonts belong in the family tree. In the later part of the 20th century, the actual bodies were found from some Lagerstätten. They turned out to have the bodies of primitive vertebrates, and indeed they are primitive vertebrates and probably cousins of today's hagfish and lampreys, that is, jawless fish of the group Cyclostomata. And so here's a life restoration, sort of a scary looking one. They have great big bulbs that are probably their eyes. They have this jawless mouth. And they've got these parts that work together, but they don't have the bones or cartilage to form true jaws. They look really scary until you consider that these things are about the size of your pinky at most. And so I like this illustration of them where they just make them look cute. Then there are the archaeocyathids. These were the first definite reforming sponges. They're a group only known from the Cambrian. In fact, they're only common in the first half of the Cambrian. They're sort of like a cone inside another cone with a series of partitions in between, and that's describing their calcite, their calcium carbonate hard parts. There are holes going through all these divisions, and that's because these are sponges. And remember the way a sponge works. The water comes from the outside, it passes through the inside, it goes up the middle, and little cells grab the food going by. In this case, the water's going both all the way from the outside to the in, but also in these other little partitions here. And you can think of this structure around here kind of like those some of those sugar cones, the more processed sugar ice cream cones you might have, that sort of structure. And here's them in cross-section. These were the first abundant reef-making organisms. Okay, Cloudina, I talked about Cloudina earlier today. They make a little bit of reefs. I mentioned that there might be some sponge reefs in the Ediacaran, but those are debated. These guys definitely make reefs. Now, the layers in which the trilobites were first found were for a long time thought to be the oldest layers with fossil shells in them. But in the mid-20th century, people were processing rocks, that is breaking them down, from in the Cambrian, but below the trilobites, in what's called the Timonian, what used to call the Timonian stage. It's no longer formally called that, and found little bitty shells. Researchers in Russia first did this, but then people started finding them all over the world, and this became called the small shelly fauna, SSF. Not very creative, but it gets the point across. Or the Timonian fauna. And, they have all sorts of little things. Some of these are probably sponge spicules. Some of these may actually be the whole shell. So these are probably the whole shell of little early gastropods, or little early snails. Maybe not adults. A larval snail starts off with a small shell and it grows as they grow up, although there were no large shells from this time. Some of these, though, turn out to be these shells that would be arrayed over the back of an organism. So it would have like chainmail on it. No one shell was large, but it had a lot of little ones. And people joked about, you find all sorts of shapes in the small shellies, like, clovers and moons and hearts and so forth. And this lecture typically falls somewhere near St. Patrick's Day. And then the Lagerstätten in Burgess Shale in Canada, Xinjiang in China, and other ones in other parts of the world have unraveled a lot more of the hidden diversity. Soft-bodied organisms. So here's the body fossil of a priapulid. Here are soft-bodied vertebrates. So things more primitive, or further down the family tree, I should say, than conodonts. They don't even have the hard tooth parts yet. Various soft-bodied arthropods and near arthropods, and so forth. A sea squirt, a group that does not normally fossilize. And among the creatures that show up, that sea squirt is an example of a chordate. So the chordates that are found in these Lagerstätten include stem craniates, or stem vertebrates, things that are outside the living vertebrates, but are still in the larger group Chordata. So here's some fossils of things like that. They show that chevron pattern muscles that we still see today in, for example, in fish. You got a nice piece of fish when you eat that. And for those of us who have them, unlike me, if you've got some nice abs, those are actually reflections of these chevron-shaped muscles. So here's a life restoration of some of these little fossils. Also, some stem mollusks. Some of these, like Wiwaxia here, have shells, in quotes, but they're shells not made of calcium carbonate, but of chitin, of the material, a very common material that's on the outside of many animals, like insects, for instance. So firm material, but not mineralized. Now, they did have some mineralized parts. They've got little radulae. So they got a radula, the scraping radula of Kimberella. In fact, they look kind of like if you had Kimberella with harder teeth, in quotes, and a little bunch of shells on the outside, but shells made of cuticle, shells made of chitin. We also have this critter, Odontogryphus. This looks actually a lot like if Kimberella was a little larger, and if its gills were a little more organized, and if it had mineralized mouth parts, mineralized radula. So the radula fits right up there. It looks ovoid shape. And it's got this gills arranged around a foot underneath and then a big dome on top. So like Kimberella did. So it's like not too far removed from that. Here's a life restoration of Odontogryphus. We even have tracks of some big shellless mollusks from the Cambrian. We don't have any body fossils of this one yet. It's quite large. I mean, that's as far across as the tire track of a mountain bike. In fact, they look like the tire tracks of mountain bikes, only these are from the Cambrian. We have stem members of the segmented worms, the annelids. And in fact, some of these have mineralized calcium carbonate plates. In fact, any one of these individual plates would be part of that small shelly fauna if you found it by itself, but we can find them also all arrayed together on the backs of these creatures. We have true arthropods. And it's a sort of a way that this seam in the rock and the light hit it, that made it like ding, like a glint, but no, it's not actually a glowing eye. So there's a primitive arthropod, a primitive member of the jointed-legged animals that today include insects and crustaceans and so forth. And trilobites. So here's a trilobite, and here's a soft-bodied animal from the Burgess Shale. Normally, we would only find this animal of these two, but you can see this animal here is just about as big, actually a little larger than this trilobite, but because it only had chitin, it did not have calcium carbonate, these guys only preserve with Lagerstätten. And here are examples of other ones that would only preserve because we have, in this case, the Zhenjiang fauna of China. And then there's this larger group called panarthropods. Probably the most famous panarthropods are the tardigrades. Probably heard of tardigrades before, the water bears, these tiny little microscopic organisms. They're more closely related to arthropods than to anything else alive. That whole clade is called Panarthropoda. Velvet worms are another group. Well, in the Cambrian, there's a whole bunch. There's like undersea velvet worms with spikes. There's weird Opabinia with 7 eyes and this long proboscis. And in particular, there are the kings of the Cambrian, the radiodonts. Radiodonts are relatively large. In fact, they're giant for the Cambrian. They're not giant from our point of view, but they're giant for the Cambrian organisms. They are not true arthropods, but they're panarthropods, and they have many different lifestyles, most of which are predatory, with big grasping limbs in front. Most famously, Anomalocaris, with these grasping legs up front, huge segmented eyes, like their cousins, the arthropods, to see them, and some of their food would, in fact, be arthropods. Anomalocaris was a trilobite hunter. And here we could see Anomalocaris itself and a smaller Lagania, looking at some of the smaller tasty tidbits of the Burgess Shale, and this gives you a sense of how big they were. They're not giants. Nothing in the Cambrian was giant compared to modern animals, but they are much bigger than any of the other animals in their environment. Eventually, the radiodonts produced forms like Tamisiocaris, which was a suspension feeder. This happens a lot, actually, in the history of life, where you get a group of swimming predators, and then they get even bigger, but they shift over to become suspension feeders, going after plankton and so forth. That happened among whales. We'll talk about the largest whales of all, are the baleen whales. It happened among sharks. The biggest living sharks are basking sharks and whale sharks. They're both ram scoop suspension feeders. It happened in the Paleozoic, among a group of jawed but toothless fish called placoderms. It happened in the Mesozoic, among some bony fish. So this happens a lot, and the radiodonts were the first one this happened to. And here's another Egriocaris, another of these suspension feeders. And here's Egriocaris, Egrocassus, I should say, shown next to a human being, and it's the largest animal known from this time. So that gives us a look at some of the biodiversity that was around in this point of Earth history, that is the Cambrian. So the biological diversity of the Cambrian explosion, most of which we would never know about if we didn't have these Lagerstätten. So if we look at the history of these things, these pick up in sort of the mid part of the Cambrian, some of these around the times of the oldest trilobites. The small shelly fossils take us back early into the Cambrian, but that first base of the Cambrian is even earlier. And down here, remember, we have things like Cloudina and Namacalathus, millimeter scale shelled organisms, and that's it. And it's looking at the diversity over time. So, what was the Cambrian explosion? Was it a phylogenetic event? Were these really the first appearances? Sorry, were these first appearances really the origin of these different groups at that moment in history? Sort of a literal reading of the fossils. Or was it a biogeochemical event, a change in the external environment that allowed creatures to start building shells and otherwise preservable skeletons? Or was it some sort of ecological event? Or was it a combination? Well, we see that there are some Ediacaran members of the crown group Bilateria. And so we saw a stem mollusk in the form of Waxia, possible stem annelids or stem panarthropods, and so forth. Well, if we have those stem members present down here in the Ediacaran, then minimum divergence time tells us that the divergence among the major groups of Bilateria had to have occurred tens of millions of years before we see the actual shelly fossils. And molecular phylogenetics, that is using DNA dating, also suggests those divergences were much earlier than the direct reading of the fossil record shows. And so we can reject the literal phylogenetic model for the Cambrian explosion. This is not the sudden appearance of these groups in terms of their actual origin. It is the sudden appearance of them insofar as our ability to see them in the fossil record. But that doesn't mean they weren't already around. In fact, we can map out which different types of organisms are showing up when, based on what sort of skeletal material they use. And for instance, here we see the stuff in blue uses the mineral aragonite, which is one form of calcium carbonate. We see the ones in red are those that use calcite, which is another form of calcium carbonate, of CaCO3. And the ones in green are phosphate mineralization. Oh, and then the in gray, those are silica. And so we see that the aragonitic forms tend to show up earlier in the record, and the calcitic forms tend to show up later. So that's the Cambrian, Precambrian boundary right here. That suggests that maybe there's something geochemical going on, particularly because each of these brackets here, those are clades. These three things are clades. So we're going across these clades, we see the same sort of pattern. We also see a pattern in terms of size, and that is we have these millimeter size calcifiers like Namacalathus and Claudina in the later Ediacaran. We had multi-millimeter scale shells in the small shelly fauna. And remember, some of these shells are the parts of larger animals, but they're making only tiny shells, just a lot of little shells in the body. And then big-shelled organisms only become present up here in the third stage of the Cambrian. That's the classic Cambrian explosion. And so it definitely looks like the acquisition of the ability to make shells is going across the family tree, but is becoming only able to make it at a tinier scale, and then a larger scale, and then a larger scale after that. As if the ability to make the shells is relatively universal for these groups, but controlled by something else. So this fact supports the geochemical model. Now, what geochemical changes are happening to control this? And I will tell you here, there is no good answer, and geologists and paleontologists disagree with this a lot. One thought is maybe it's just oxygen. Maybe it's the oxygen level just has to get higher and higher and higher. Other people have suggested maybe it's some other change in water chemistry, maybe something with the acidity, maybe with the relative proportion of phosphates versus calcium. Maybe it has to do with a ongoing shift, as happens again and again throughout Earth history, in terms of the intensity of the mid-ocean ridges. There's a bunch of different things that it might be. Definitely, there is a great increase in marine oxygen and in atmospheric oxygen at around this time. Why should that happen, though? There's two main thoughts as to why it might be. One is that the increase of small and medium-sized animals from sponges on up in terms of complexity as suspension feeders have begun to clean up the water column. Maybe the water was sort of murkier before with undigested organic material, but now it begins to get cleaned up. And as it gets cleaned up, instead of just being controlled mostly by primitive cyanobacteria, blue-green algae, now it's eukaryotic algae, complex algae, that become better able to use the sunnier sunlight reaching the water, and they're more active and they put out more O2. That's one possibility. Another possibility is that maybe there's very primitive land plants as well as lichens colonizing the land. The land plant evidence is mediocre. They're evidence of spores of the type of green algae from which land plants evolved in the Cambrian, but we don't know if those are actually creatures living on land or just in the fresh water. And lichens, it looks like they probably have colonized land by that point. Lichens are this weird assembly of fungi and some algae together. In any case, if we now have parts of the land surface producing oxygen, that's a brand new source that wasn't there before. And so here's this idea that Proterozoic oceans and Phanerozoic oceans were different, that modern oceans with their complex food webs are well mixed, they have clear water, they're dominated by eukaryotic algae, as opposed to the dirty water, the simple food webs, and low oxygen levels of the Proterozoic ocean. It's an interesting thought. This figure should have been a lot earlier, but I can use it now. This shows the size of Cambrian animals. And we could see here that the very large omnidens, which is the largest radiodont, is about human-sized. The other radiodonts, much smaller, and they're still much bigger than other animals at their time. These are other types of animals, and they're repeated down at this level. So yes, the world of the Cambrian did not have a lot of big animals. One thing I didn't address yet, though, is why biomineralize? What's the point? Why build a shell? Well, the first thing we'll think about is defense. That makes sense. A lot of animals use their shells for defense. That's certainly true. And indeed, it's a defense that predators have to build a response to. Like we saw the little hole drilled in Cloudina, that showed up after the appearance of Cloudina. But shells aren't always good for defense, or only good for defense. If you're a very tiny animal, you could just get swallowed anyway. Another important aspect of making a skeleton to biomineralize, it's the mineral store. That is, if you put this extra minerals outside your cells, but within your body, then you can dissolve them back up and use them for your own physical function. We do that, for instance. Our bone is constantly being resorbed by our cells and then replaced by our cells again because we need that calcium or we need that phosphate to do stuff. Also, a very common issue for a lot of organisms is the need to get rid or dump excess calcium. Why not build a shell while you're at it? And then if you've already begun to build a shell, it turns out that having a skeleton is a good way of getting stronger because now you've got something to act against. I mean, it seems obvious to us. If we didn't have an internal skeleton or we just had the muscles, we'd be flopping around. We couldn't do anything. But with muscles around a bony skeleton, we've got levers and a power source to drive those levers. Well, the same thing happens with a shelled organism. They use the shell as something their muscles can tug on, and now they can be a lot stronger than they could before. So there's many different benefits. And if you can start to do more, you can have more complex food webs, more complex ways of doing things. And we see, compared to the simplicity of the Garden of Ediacara, more complexity in the Cambrian. You can now have multiple layers of predation. So not just grazing and the creatures that eat the grazers and the suspension feeders, and that's it. Now you get the creatures that eat those creatures and the creatures that eat those creatures. You can begin to get pelagic communities, that is, communities that are swimming around, nekton and plankton, and more complex infaunal communities, burrowers. So the whole food web gets more complex. So What was the Cambrian explosion? Not, as traditionally thought, the sudden appearance of these many groups per se. Instead, it's likely that it's a biogeochemical event driving an ecological transformation. So the seas change in some fashion that allows the building of shells to be easier. Once you're building a lot of shells, then wow, you can interact against those shells and live in all sorts of new ways. And that will drive an ecological explosion of form. Oh, and I should show here on this diagram, you see this blue to red shift here. That's when ocean chemistry changes in terms of whether it's easier to generate aragonite as the carbonate material versus calcite. And this is not a geochemistry class, so I don't want to go into great detail. It has to do with the activity at the mid-ocean ridges. It does seem to coincide, the switchover, with the first appearance data of the big macroscopic forms with shells in the Cambrian. So that's one reason many of us think that's ultimately a biogeochemical event, an extrinsic environmental change that drives a major biological and ecological transformation. So in other words, it wasn't just a single boom, but rather there was, as I say, a long fuse to the Cambrian explosion. Step-by-step changes among various forms, diversification, although mostly at a very tiny body size, but when the environment begins to change right, organisms, animals are there to take advantage of it, and once they become these shelly animals, they start interacting with each other in all sorts of new ways. Although, we'll give you an alternative hypothesis here, which is somewhere in Cambria, by the way, that's Wales, there's an explosion and the radiation causes all sorts of new forms. However, don't use this for your report because you will get an F. Or, finally, another alternative hypothesis. Well, take care, and I will see you in class on Tuesday, so we'll talk about trilobites and ammonites. Take care.